Alright, so we're going to go learn about mutations, and what we first want to do is Google the right web page. And for this one, we're going to Google the term or the phrase molecular workbench mutation activity 102, as you see right here. And the first option you see is what you want to select. Changing the genetic code to 7-page activity by the website called Molecular Workbench. And so here we go. This is it. When you go to this website, all you have to do is click the shot once, and it'll tell you please just click it once, and of course do that so it loads properly. And if everything goes well, as it's been with the school laptops, we just want to hit run. So let's get rid of that. Alright, so what we want to do is click on this third chapter here, how mutations work, substitution mutations, and click on it. And as you can see, it says how mutations work. Now it says substitution mutations here, but we actually have the ability to do any number of mutations with this activity. Alright, now that we're here, let's review the worksheet, and let's kind of figure out how to use the worksheet with the activity. So with an activity like this, we first want to identify the problem, and just with any good research with science, that's what it starts with. So we need to ask ourselves a question, what's really motivating what we're doing today? And that is, how do mutations affect the DNA or making proteins? Right, so you can, when you do the activity, have this specific problem, or so you can borrow it from this, or you can come up with your own. What you can see here is also, as we've done before, it's asking, what did you see happen? So what happened, or what are observations you notice? And for this one, we could have up to 7. You may need more, you may need less, just based on what you choose to write about. And in this space, also, these observations 1 through 7 can also be drawn as well. So things you see that you want to draw, feel free. And I'll talk about that with the website. But anyhow, our goal is really to play with the activity site and then just write what we see, what we've learned. Let's start it off first is just you can transcribe DNA to RNA. Let's see what happens. All right, so we have the DNA strand here, the DNA strand here, and we have this new middle strand made up of nucleotides A, G, U, and C. And as you can see, I go down here, it'll tell you if it's RNA or not, so that's kind of interesting. It's actually telling you with the red band here, this middle strand of nucleotides is actually, it just tells you right off the bat. So we basically made an RNA strand from the DNA. And then you click here, which could be an observation. So if you wanted to go back to your first observation, you could say, for example, DNA strand during transcription led to something called mRNA being produced. It's just an example. It's all up to you. Alright, so let's go back to the website. So we did that. Then we go to translate and see what happened. And you can see these circle shapes bonding together, forming a new structure. As we've studied in the past, we know that DNA eventually makes a protein. And so this kind of ties back to the original problem we had, which is, and it's basically DNA's involvement with making proteins. All right, but let's go. We still don't know how mutations are involved, but this is what we do. If you take a snapshot of this, you can hit this button. it takes a picture of it, and you can actually see which amino acids and in what order is produced from the original DNA strand. With the mutation, we're going to do something different, and I'll show you different ways you can mutate the DNA. But you really need to make snapshots of each protein you make so that you can compare it to this original one. And that's really important. Otherwise, it may be difficult to know what the mutations did. All right, I'm just going to hit OK, but always the snapshot function is there. So let's repeat it one more time. You can always reset, and we'll need to do this throughout your activity. So you just hit yes, I want to reset it. And again, you just hit transcribe it, then you translate it, makes the protein. One bit of advice is that you may want to click it soon before it folds up into a more complex structure, so it's more easy to see the leading one. And if you don't do anything to the first 3 series of nucleotides in the DNA, you'll always have it start with ARC, so it's a good placeholder for you to know the first, second, third, fourth, fifth. My advice to you, of course, is to actually draw or document which amino acid is which, just as you see it here, for better comparisons later. All right, so let's get to the let's exit out of this. So we're going to reset one more time. Again, the point of this, so you'll have to reset each time. The point of this is to really learn what mutations do. And you right-click, so instead of a left-click, you right-click any of the nucleotides here, any of them, and it pops up 3 different ways you can mutate the DNA. Let's start with substitution. All right, and what just happened is we replaced one nucleotide for another. We didn't remove any, we didn't add any. All we did was change one nucleotide from what it once was. And so I'm going to reset that one more time, just so you can see that one more time. So you right-click any of these. Let's right-click this C nucleotide here on the bottom. And let's do substitution. We're going to change the C to an A. And you can see that right here. So this was a C and an A, now it's or it was a C and a G, now it's a T and an A. Anyways, we mutated. We changed a nucleotide from a C to an A. All right, now let's transcribe it. Boom. And then let's finally hit translate the RNA to protein. And I'm going to take a snapshot real early on. Boom. And then by taking the snapshot, again, you can look at the protein produced and really just document, either in writing or drawing on the observations, the order of the amino acids and the name of each. Again, this is important because you want to compare the mutation we did with the original unmutated DNA strand and how it kind of impacts the protein or how it or what a mutation does to the DNA. So let's kind of discard that, and it's floating around there. It'll continue until you reset it. Let's look at the worksheet. So here, any observation, your observations could focus on what a substitute mutation did to the DNA. An observation could be based on, let's see, what the protein looked like, or the amino acid sequence of the resulting protein. So really just focus on what's happening to the DNA based on the different mutations you do, or what the proteins themselves look like from the original protein, from the unmutated DNA, or proteins made from different mutations. Again, just to share, let's look really quickly again and just remind. So let's reset it, because you got to just do a series of resets. Reset it again. Let's right-click the same C we did before, and instead of a substitution, let's do an insertion. And what this is not turning a nucleotide into another nucleotide. It's really just adding a whole new nucleotide to it. So there's one more nucleotide than there once was, and you can see from the full length, there's one more. The DNA strand is a little longer than it once was because you added. All right, remember that is a insertion. Yeah, insertion mutation here. And then deletion is the opposite. If you click deletion, you really just deleted one of the nucleotides. So the strand is back to the same length, but the nucleotide I deleted was different than the original strand. And when you do that, again, every mutation you do, or every different mutation you do, you can document each form of mutation, and you can take a snapshot of the resulting protein and really decide what kind of effect is each mutation doing to the DNA. And the best way to do that is to truly document in written or visual drawing what the amino acid sequence is, and what the overall protein looked like. All right. So after you get all your observations, your job then is to come down to this middle box here and add a hypothesis. Why do you think things are happening? So provide a confident explanation or a claim of why you think it's happening. And then here at the bottom, you have a choice of, here at the bottom you have a choice of, right here, you can either write a kind of summary of what you've learned and the evidence supporting what led to your hypothesis here, or you can draw a model, or some visual model, if you want to represent your thinking and what you observed that way. That's fine. If you do choose to make a model, just remember some of these key details, such as the model itself really should reflect the questions you're asking about the DNA, such as your original problem, which was, so it should show your thinking on the question, how do mutations affect the DNA or make a protein? But if it doesn't do that, it can also show your best understanding of the events, so what you best understand from what you saw. And of course, with any writing or model, it's you showing your first draft level of understanding. So just assume we'll change it, we'll learn more, and adjust the model or the writing as needed. It can reflect your hypothesis from the middle box, which is this section right here. And given that it is a process, mutation itself, should be able to show some kind of process before and after, so before the mutation and after the mutation, or what caused the protein or the amino acid sequence to change, etc. And don't forget also to add symbols or a key to show which thing is which in your drawing, if that's what you choose. All right, good luck with the assignment.